All right, good morning. Welcome to Pediatric Grand Rounds. I have the pleasure to introduce upcoming Grand Rounds and then introduce our speaker today, who is 14th annual Cohen. What number Cohen lectureship are we up to anyway? What number Cohen? What number? I know. I was asking, what number lectureship are we up to? 11th annual Cohen lectureship. So again, I'll introduce today's speaker, and then I hope all of you have a happy holidays, and then we'll resume January 14th with Dr. Glowing, who will talk about lessons from human genetics for diabetes from bench to bedside. And then on the following week, we have another one of our now recurring series of House staff debates, and the title of this debate this House believes that scheduled feedback is a valuable use of our time. Yeah, will please attend. We have a number of really interesting upcoming workshops and days and symposia, including the Stanford Pediatrics Engage program, which you can see here. And then we have a program on echocardiography course on congenital heart disease in April and May. And so today we have, again, the 11th annual Cohen lectureship. And Harvey, this lectureship is to celebrate Harvey's contributions to both LPCH and the field of pediatrics and in general, after being the chair and chief of staff here for a number of years. And what Harvey asked for was, instead of honoring a distinguished career investigator such as himself, he wanted to use this as an opportunity to highlight rising stars and junior investigators who will one day become the senior distinguished investigators like Harvey. And so with that, it's really been exciting to use this lectureship not only as a way of highlighting and potentially recruiting junior investigators from other institutions, but it's been our opportunity to highlight the new hires here at Stanford. And today we get to use that by honoring Natalia Gomez-Ospina as the Harvey Cohen lectureship. Natalia is a MD Ph.D. She did her MD and PhD training here at Stanford. She did her graduate work in neuroscience and the physiology of brain development under Ricardo Gomez, who's now in charge of the neuroscience program at Novartis. She then took a little while to figure out she wanted to become a pediatric medical geneticist and took a little foray through dermatology, but kind of indicating her productivity in that little brief one-year foray in dermatology. She ended up publishing a first author New England Journal paper. But she realized dermatology wasn't really exactly right for her and found her way back to pediatric medical genetics, where she trained here. And then as part of her postdoctoral work, worked in my lab, where she will describe the work she did in terms of using the blood system as a way of delivering proteins and therapeutics to treat neurodegenerative diseases and brain diseases. What I will say is Natalia is a fantastically creative, rigorous, and productive scientist. And in addition, she's a fantastic awardee of this award because she also takes seriously the idea of mentorship, not only of people who already know what they're doing, but people who still need to know what they're doing. And that takes a lot of work. But Natalia has shown a tremendous aptitude for training people and turning them into really rigorous, innovative, and productive scientists like herself. So without further ado, Dr. Gomez-Ospina, please, welcome you.
Thank you, Matt, for the introduction. Thank you all for attending, and thanks to my benefactor, Dr. Cohen. I thought I would start this talk by telling you what I'm not going to talk about. And I am not going to talk about germline genome editing. I know some of you want to talk about this. I think this is going to be the topic of a future Grand Rounds, and this guy here will be suited to talk about this. So everything we're going to talk about is germline genome editing. So I'm going to talk about a lot of things. Some of them I might not explain very well, but what I really want you to walk away knowing is these things. So genome editing based technologies are here. People are being treated and you really need to understand them. So far, it hasn't been very long, but they appear to be safe. Efficacy is yet to be determined. Some trials are looking very promising, some are not. And hopefully you'll see that there are actually several ways to approach individual diseases. So you'll see that, here. This too? Okay. All right, guys, stuck here. So there are different ways to approach an individual disease, and so you'll see. I'm going to give you an example of that, and hopefully you also learn that you can use the blood to treat multisystemic diseases and specifically diseases of the brain. So the real overview is we're going to go over the genetic modification tools and discuss a little bit some of the pros and cons. There's actually several of them. So I want you to at least know these names. We'll talk about an example of treating non-hematological diseases, specifically lysosomal storage disorders. And discuss the two ways of approaching them using genome editing, in vivo and ex vivo approaches. And we'll focus primarily on the proof of concept studies that we did to use ex vivo editing of hematopoietic stem cells to treat LSDs. We'll also expand on how we're using this platform to treat other lysosomal storage disorders like Krabbe and Gaucher, and we'll summarize some take-home messages. So in truth, genome editing technologies have the potential to impact many areas of human health, not necessarily disease, but I'm a medical geneticist, so I focus on this problem, and this slide is here to remind you that genetic diseases are an important health problem. There are millions of people affected in the US, hundreds of millions worldwide. Most of these are children, and most of these children will die of their genetic disease before the age of 5. There are very few FDA-approved therapies for genetic diseases, but even fewer of those are sort of one-time and definitive therapies, which is kind of what you really want to do if you can. So this is my bird's eye view of gene therapy. I know that you hear this term a lot, and this is how I think of it. I think of gene therapy as a way to deliver. It's the delivery of genetic material into cells, and I qualify that as a long-term delivery of genetic material. And there are broadly 3 different ways of doing this. You could do it with non-integrated viruses like AAVs, and so you'll hear about the use of gene therapy using AAVs. You could also do it using integrating viruses, and so this is what I call non-targeted gene addition, and you'll hear, there are people here who've done a lot of work on this, and there's genome editing, which allows you to make very more precise modifications. There are actually multiple platforms. Everybody says CRISPR, 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 but in reality, there are multiple ways of doing this, and some of them actually made it to clinic before CRISPR. So people have actually been engineering cells, and primarily hematopoietic stem cells, for decades. And so people have been using recombinant, non-replicating viruses to permanently alter human DNA. And so those early trials had some adverse effects, but people went back to the lab and made safer vectors. So in truth is that now gene therapy using integrated viruses, retroviruses, and lentiviruses have established very good therapies for some genetic diseases, some that are hematologic and some non-hematologic. It's important to, even though this talk is all about genome editing, gene therapy using viruses is still very relevant, and there will be some therapies that hopefully we'll be delivering in this hospital that use viruses. So despite great progress in viruses, it sometimes is better to be able to be precise in the genetic changes that you make. And so genome editing is a tool that allows you to very precisely edit DNA sequences. And so most people think of genome editing as a way to take an incorrect sequence and turn it into a correct sequence. But what I'm here to argue is that genome editing also allows you to create new sequences that sometimes have more therapeutic value. So as I mentioned before, there are multiple ways of doing genome editing. Most of the ones that people are using nowadays for therapeutic purposes are these based on this nuclease-based technologies. And at the core of all those base technologies and at the core of being able to make a very precise genetic change is to be able to create a double-strand DNA break exactly where you want it. And so what the different technologies are going to differ in is how they recognize that DNA sequence. So for example, these were sort of the early versions of what I call it engineered nucleases. They have a fusion of nuclease. It's called a Fock nuclease, which I'm showing you here with scissors. And they recognize DNA with this different protein motif. So on the left, these are called the zinc finger nucleases, and they recognize DNA using an array of zinc finger DNA binding motifs. And here, these recognize DNA using another series of proteins called transcription activator derived from plants. But so among these two sets of engineered nucleases, the zinc finger nucleases actually were the ones to make it into clinical trials before any other platform. So we'll talk a little bit more about them. Now there is a third generation, the one that you probably hear the most about, which is the CRISPR-Cas system derived from bacteria. Technically, what we use for basic research and for therapeutics is a engineered version of this bacterial system. And the system really has two components: a guide RNA, which some people describe as a gRNA or sgRNA. And the guide RNA is going to guide the nuclease to the sequence you want to cut. And that nuclease is called a Cas protein or CRISPR-associated protein. And most of the proteins that we use in the lab right now are called Cas9s, and they come from Strep pyogenes or Staph aureus. They've been evolved to be synthesized in eukaryotic cells. So you can imagine that if the only thing you need to do to change the sequence that you target is to change just an RNA sequence, is actually really easy to do. And this is the reason why everybody is using CRISPR. It's quite versatile, it's cheap to engineer, and so it's been now widely adopted both in basic research and in therapeutic development. In the future, and probably now, you'll hear about base editors. These are based on these nucleases, but they don't cut DNA. What they are, they're fused to these base-modifying enzymes, cytidine deaminase or adenosine deaminase. And so what happens is they bring this DNA, the modifying enzymes, and like a cytidine will turn a C into a T, and the adenosine will turn adenidine into a guanine. So it allows you to mutate those specific residues within a very specific binding site. So it's great. You don't have to cut DNA, so presumably that might be safer, but it's sort of limited to if those are the residues you feel that you have to mutate. So there are some limitations on how you use it. So everything, as I mentioned, starts with a break in the DNA. So what happens after that? So the cell is going to try to repair that break. There are two ways in which it's going to do it. It's going to use a mechanism shown here on the left. It's called non-homologous enjoining. And so usually that works pretty well. The cell will be able to repair it. But when it does that, it regenerates that target sequence. And if the nucleus is still around, it's going to get cut again, regenerated, cut again. Eventually, there's going to be a mistake, and generally those mistakes look like small insertions or deletions, which we call them indels. So NHEJ will generate indels, and those indels are hard to, and they're kind of, you can't really predict what they look like. They can be very variable for each guide and in each cell. So they're hard to predict, but it can be useful for therapeutic processes. A better way is to use homology-directed repair, which allows you to make very precise modifications. So what you do is you make that break, and you give the cell a template to repair that break, and that template is homologous to the area that you cut. And so with this technology, you can actually make a variety of genetic modifications. And so an example here is you can take a single nucleotide mutation and change it back to a wild type. And this is a great approach in diseases that have a common, for genetic diseases like sickle cell disease, where everyone has the same mutation, and it's quite efficient. Now, most genetic diseases are not due to a single mutation. They're due to mutations that are spread out throughout the coding sequence. So you can instead use this approach to insert a healthy coding sequence within the gene and maintain regulation. So this becomes a one-time strategy for all pathogenic mutations in a disease. Now, there are limitations to the size of this repair DNA molecules that we can put in cells. So for some large genes, this isn't feasible. It's just right now there's a limitation to the size of this. So what you can do is maybe you can exchange exons or multiple exons. Generally, you go for areas that have multiple, there are mutational hotspots, and some diseases have that. What I think is a more interesting thing to do is to actually insert a sequence that has a promoter you design and a sequence you design. And generally, these are better if you stick them in a locus that we call a safe harbor. So we take an entire a different place in the genome and put in a different gene that we completely engineer. And so this safe harbor is a place in the genome that we think is not necessary for the function of those cells or the function of that organism. And I'll give you an example about that. You could also add sequences to your proteins, and in basic research that can be very useful. You can use it for visualizing, localizing, watching proteins move around in cells, but you can imagine that sometimes you can add sequences that may enhance some function of a protein, like help it cross the blood-brain barrier, for example. That's what I'm interested in doing. So my father-in-law is a retired lawyer, and he took a class on CRISPR, and he's like, that sounds great. You can change DNA, but how do you really treat a person with this? And that's an excellent question. So in order for genome editing to be therapeutic, you have to be able to put it in the right cells or in the right organ systems, correct? And there are generally two approaches to do this. There's the ex vivo approach, or outside of the body. And for you to be able to do this, you have to be able to isolate cells, modify them outside of the body, and they have to be transplantable. So your organ system has to have that type of cell available for you to do that. The in vivo approach is you just stick these things into people directly and hope that these vectors carrying your genome editing tools will get to the right organ in the right amount. So for when you're trying to target the hematopoietic system, it makes a lot of sense that we use an ex vivo approach. We have decades of experience isolating the stem cells, modifying them and transplanting them. And so the advantage of going this route is that, number one, it allows you to look very carefully at your genome editing outcomes, because while you may make your intended modification, there's a possibility that you make unintended modifications. So it's nice to be able to do this outside of the body, so you really evaluate the safety of your system. You can also better evaluate efficacy because you can really measure the phenotype correction in these cells. And so for hematopoietic based therapies, of course, we are using an ex vivo approach. So here are some examples of non-hematological diseases that I think can be very treatable using the blood system. There are already other approaches using genome editing, but I'm going to give you an overview of this condition. So lysosomal storage diseases, hopefully some of you remember, these are the ones that you chose not to learn about in step one, that was me, are a large group of genetic conditions that are due to deficiencies in these proteins that are important for lysosomal function. Lysosomes are these very important catabolic centers within cells where we degrade and recycle big macromolecules. And so you can imagine that this ensyncerator function is important in many different cells. And so when this doesn't work very well, there are many different cells that can show problems. And this is the reason why these diseases show manifestations in multiple organ systems. Cells in the brain, like glia and neurons, are particularly susceptible to lysosomal dysfunction. So many of these lysosomal storage disorders have neurological involvement. And some people would argue that, in fact, adult neurodegenerative diseases are due to sort of low-grade lysosomal dysfunction. So these are a large group, as I've mentioned, more than 50 disorders, but most of them are due to problems with these enzymes that are within the lumina of the lysosome. Not all of them are enzymes, some of them are membrane proteins, but the beautiful thing about these enzymes is that they're constantly being secreted into the extracellular space. And they are modified with these sugars that I'm showing here in red called mannose 6-phosphate. And they can be internalized by any cell that has a mannose 6-phosphate receptor. So what this means is that a cell that makes enzyme can correct a cell that is not making enzyme. And just by virtue of letting some enzyme diffuse into the vicinity. And this is a property that is known as cross-correction and really is the basis for creating all these therapies for LSDs. So the model LSD that we're going to talk about is this disease called mucopolysaccharidosis type 1, or MPS 1, also known as Hurler syndrome. It is our model disease for our therapeutic approach. It's a canonical LSD. As you can see from this table, it affects many organ systems. Like many enzyme deficiencies, there's usually some degree of variation depending on how much enzymatic activity, residual enzymatic activity a patient has. So if you have almost nothing, you have severe disease, which the most important manifestation in severe in Hurler syndrome is neurological deterioration, which if untreated it's lethal within the first or second decades of life. If you have some enzyme, you actually get to have pretty preserved cognitive function. There are some subtle differences, but these patients are, quite different in their natural history. And I'm showing you here the median enzymatic activity of these different patients, so almost zero, and you get bad disease. Even with a 1% activity, it makes a big difference. And this is important in knowing when you're trying to define, all right, how much enzyme do I really need to give back to a patient in order to really make a difference? So we're not shooting for 0.79%, but that also means we're not trying to shoot for 100% either. And so if you say, well, that's a really weird disease, I don't know what you're talking about, it may actually impact you because MPS 1 is now part of numerous screening programs in several states across the United States. The states shown here in blue and now in our home state of California. And I believe our first local severe MPS 1 is being transplanted at this institution right now. So the disease is due to a deficiency in this enzyme hyaluronidase. When you don't have it, it leads to the accumulation of these giant sticky molecules called glycosaminoglycans. We call them GAGs. And too much GAGs, and you have a progressive multisystemic disease that is bad for your brain. So these patients can be treated with enzyme replacement. This is the weekly intravenous infusion of a recombinant enzyme. So you can imagine this is onerous, it's extremely expensive. It doesn't work very well. It doesn't cross the blood-brain barrier. So if you have severe disease, it's not going to help you, and it doesn't really treat the bony manifestations of the disease as well. So it's a therapy for patients that have mild disease. And we also do allogeneic hematopoietic stem cell transplants, which generally works better. But you can imagine this is a risky procedure, and because of this morbidity and mortality, it's only reserved for patients that we know are severe, they're young enough to undergo it, and they have enough preserved cognition that we know it's worthwhile taking the risk of giving them a transplant. So as I mentioned before, there are two genome editing approaches to treat these diseases. So approach number one, it's the in vivo delivery of genome editing technologies using stem finger nucleases. This is what other people, a local company is doing. And so what they said, why don't we stick the enzyme into another gene? So they're going to put it into the albumin locus. And so the hope is that because the albumin albumin is normally highly expressed in the liver and secreted, you can actually trick the liver into making enzyme instead of albumin, and some of that enzyme will be produced by the liver, and that will help some organ tissues. And this is an in vivo approach. People get injected with this nucleases and the templates. And show of hands, who thinks this is going to be safe? Don't think about it too much. Are you afraid? Yes? Does it sound like it's going to work? Yes? All right. It's actually happened. So this is actually the first human being that underwent genome editing for one of these therapies. So there are two ongoing clinical trials. The one most relevant to us is the NPOWERS trial, which is a phase 1 clinical trial for using this approach for MPS 1. The interim analysis, and this is another trial for MPS 2. The approach is exactly the same. Stick it in the albumin locus, have the liver make the enzyme, and hope that at least that gets them off the ERT, which is, I think, is pretty reasonable. The interim analysis says, no serious adverse effects. It's been a couple of years, great, but they haven't been able to quantify any enzyme in the blood. So what's happened so far is that they weren't able to get enough modification in the liver to actually get a quantitative effect. So, yeah. It's been done, and the data is still being collected, and I think they're trying to modify the approach to get more modification. So there's still at play, might be an option. But if we go back to this, we said, why don't we leverage the ability of the hematopoietic system that we already know works and make it a better therapy? So why does hematopoietic stem cell transplants work in helping other things besides bone marrow? And the reason is that you have the stem cells and the progenitors in the bone marrow, and some of these cells are going to become monocytes and macrophages. And the role of these cells is to migrate to injured tissues and become tissue-resident macrophages. So what we do is we rely on these cells to be enzyme delivery machines. And they have tricky ways to get into the brain. If you give them the right signal, they can get into lung, they can get into spleen, and they can get into liver. So, and how they do this is something that we're trying to understand, but it has a little bit to do with the condition that we give them during transplantation, and it has something to do with the process that is going into in the organs that sort of saying, hey, come and help me, I need some macrophages. So we thought that we could improve the therapies for MPS I and other LSDs by saying, why don't we take the patient's own hematopoietic stem cells, which they're hematopoietic stem and progenitor cells, HSPCs. So HSPCs from now on, we engineer them outside of the body, and then we give them an autologous transplant of their own cells. So there's less risk of graft versus host disease. You'll always have a donor. We'll get to treat it faster. And then the idea is that you transplant them, and then these will make the macrophages that we're relying on to get the therapy, throughout the body. So instead of doing mutation correction or trying to engineer the iduronidase locus, we said, why don't we try safe harbor locus? And so back to the safe harbor question, again, safe harbor is a place in the genome that we think you do not need. And so there actually are such genes, there are people who have loss or homozygous deletions of certain genes, and they're perfectly healthy. So an example of this gene is CCR5. About more than 10% of the European population have homozygous deletions in this gene, so they don't express it. And the way that it was discovered is because without this receptor, you're resistant to HIV infection. So we're pretty comfortable saying this is a locus that we can repurpose for the purpose of making enzymes and perhaps even other therapeutic proteins. And so the beauty of using as a harbor is we do not have to design the therapy every time we get an MPS I patient with a different mutation. It actually, for these diseases, we know that transplants aren't perfect. They're relying on the little amount of enzyme that a normal hematopoietic or normal bone marrow cell makes. So we can further engineer this to have the bone marrow make more enzyme or better enzyme. So it gives us the flexibility to do that. But most importantly, it's really flexible, and it's a platform for switching enzymes, promoter sequences, and even if you're not interested in lysosomal enzymes, you can think about expressing other things that could be useful. So their approach versus our approach. So what happens, here you're relying on the liver to make the enzyme. It's not going to get into the brain. It's not going to get into the bone. Whereas this approach is actually going to be able to do that. It does require that you get cells out of the patient and transplant them into, so it's a little bit more involved. But I think in terms of treating severe disease when there's neurological symptoms, this is going to be a better approach. So these are the experiments that we have done, and I'm going to review with you very quickly on how you demonstrate that your hematopoietic therapy is going to work. So we isolate the cells, either from cord blood or peripheral blood. The cells are modified right now we use CRISPR to modify them. And then we measure, how efficiently was our modification? Are we expressing how much of the enzyme? Are we expressing how much of the enzyme? Are we secreting? How we impacted the properties of the stem cell? Because that's really important if you're trying to design a one-time therapy. Are our stem cells still stem cells? And then, and how do we assess the phenotype correction? How do we assess efficacy in this situation? So we use the Cas9 protein, a single guide RNA. We deliver them with electroporation, and we're creating a double-stranded break in the middle of the CCR5 gene. So we're breaking it, and then this is a template that we're going to give the cell. It's a schematic of a sort of generic template for repair. So it has some promoter that we get to control. It has an enzyme that we actually get to control, and you may add a selectable marker if you feel like, oh, maybe I need to select the cells, or maybe I need to visualize the cells, which is what we're going to do for the mouse study, is not what we're going to do for humans. So how efficient is this? So here is a couple of ways of modifying the cells. So here are cells that are expressing this particular enzyme at pretty high levels. Here we used a fluorescent protein for our preclinical experiments to be able to track these cells in mice. So the overall efficiency, each one of these dots is a human donor. So if you repeat this experiment a bunch of times, 35% of the HSPCs that you can get from a person can be modified. And if you make one of these smaller modifications, you're removing this, then it gets more efficient. So you get more like 45% of the cells can be modified. So it's not perfect, but it's efficient enough that we can say we have enough cells to try to see if this works. Now these cells, we can recognize them based on the expression. They are expressing this marker that we give it. It makes it very easy to do the experiments. And so they're high for the fluorescence, and we know that these cells are modified because if you culture them for a long time, they'll always express it because this thing is in the DNA. And these cells, as I mentioned, our goal was to make cells that were better at making an enzyme than normal cells. And so these cells make 50 to 25-fold more enzyme than a normal bone marrow cell. And, as I mentioned, these cells are released in this extracellular space, so we can actually measure them in the media. As I told you before, central to this whole approach of using bone to treat solid organs is the idea that you are going to make macrophages that can still do this. So we can take those cells and show in a plate that you make macrophages. These macrophages are still secreting tons and tons of enzyme. So an important question about genome editing that a lot of people talk about is, what about unintended cuts in some other places? What about unintended modifications? And people often refer to this as off-target modification. And so there are different ways to look at this. You can do, people do this so we're not going to assume anything, and we're going to look everywhere in the genome and different ways of doing this. What we're doing here is we do a biased bioinformatics guided approach. So we look for other places in the genome that have a sequence that look like the sequence we're targeting, and we're trying to see if we made cuts there. And so by doing sequencing. And so we predicted that there could be 67 sites, and then we sequenced them deeply to see if there were modifications there. And 4 of those 67 sites ended up having what you would call a true off-target, meaning they were above the limit of detection for this technique, and they were above background, and they repeated in a couple of different experiments. They're still pretty low. Less than 0.5% of all alleles had a little something in there. But the most important thing is that if we switch to a new version of the nuclease, which has been evolved to have higher fidelity, those things with the blue go away. So as far as we can tell, we're not making unintended changes in other places in the genome. So if this is a one-time therapy, you really have to assess that the cells are still stem cells. And so for those of you who don't work with this is sort of an image of the hematopoietic compartment. You have a stem cell that can self-renew, and it maintains all of these cells, and that cell is supposed to be able to give rise to all these lineages. And so we're going to try to show that our modified cells maintain this ability to self-renew and are capable of making these other cells. And the standard of way of doing this is to put cells in mice and watch them engraft serially. So you put them in a mouse, you wait for months, you check to see if they're there, and you take them out, you put them in another mouse, you check for another months to see there. So this is what we call serial engraftments. If you keep seeing progeny of the cell after 8 months, that's sort of how we think these cells probably have long-term regeneration capacity, and we're probably dealing with stem cells. And so here, which I'm showing you is after 4 months, here an example of cells where we actually selected them based on this reporter. We looked for fluorescent cells. Again, we can track them. After 16 weeks, they're there, and you can see that after 16 weeks, they're still expressing. So we know expressing the marker, the fluorescent protein. So we know the cells that are in the mice at the end of all these months are modified. There's a little bit of a decrease in the engraftment potency, and that has to do with the fact that this modified population probably has fewer long-term repopulating clones, because the process happens more efficiently in cells that are cycling and not in the stem cells, which tend to be more quiescent. So how do we show that these cells are effective? And targeting human cells is great because eventually that's what you want to do for people, but it does pose an important problem when you're trying to show efficacy, because normally you show efficacy of a therapy in an animal model, and then you have to contend with the immune system of that animal model and deal with rejection. So you kind of have to go back and remake your model system. So we have to make models of these diseases they're immunocompromised, so they're not going to reject our human cells. And so these models, immunocompromised models of the diseases, replicate the disease well. They don't have enzyme activity. They have GAGs, like I mentioned, the patients have, and that have bony and visceral manifestations, as you would expect for patients and mice that actually have intact immune systems. So what happens when we transplant the cells? We have beautiful biochemical improvements. So you get reconstitution of enzyme activity in the serum and liver and spleen, and even in the brain. You get, you know that you're decreasing GAG storage throughout the body because the urinary GAGs go down, and then we're decreasing tissue storage of GAGs, particularly in liver and liver and spleen. We didn't show in brain, but we think we can show that now that we have better engraftment. Accordingly to these biochemical improvements, they show phenotypical improvements. The bones are thinner, longer, the way you'd expect them, and in neurobehavioral studies, they also perform better. So here you're comparing treated mice, which are the red lines or the red bars, to untreated mice, knockout mice, that are the blue lines, and you can see that these mice are walking around more, so they have more spontaneous locomotion, they're rearing more, they are remembering better in short-term memory tests of passive avoidance, and they have more of a normal digging. Some people think that digging behavior is related to anxiety. So hopefully you believe me that we can engineer this human cells with relatively good efficiencies, that these cells maintain their stem cell potential and their potential to be definitive therapies after the modification, and that for this particular disease, these cells are capable of improving phenotype in mouse models of the disease. So when I described this to you, I described it as a platform, and so that's what we're trying to go after. So as I mentioned, you can switch promoters and enzymes very easily, and so ideally you could go after other enzymes within the lysosome and try to use this to see if it works. So so we're using this for Gaucher disease, and this is work done by who was then an undergraduate student in the lab and now is an MSCP student. The example that I gave you is we tried to make a lot of enzyme, and so we use a constitutive promoter. So it's turned on in every cell all the time. And the goal was like, let's make as much as we can. And we knew that it wasn't going to be bad for the stem cell as much as as far as we could tell. Sometimes though, this is a very artificial system, and like not all cells would like to have to be overexpressing tons of a lysosomal enzyme. And this is particularly true for the enzyme deficient in Gaucher disease, glucocerebrosidase. So we learned that if you put it in the hematopoietic stem cells, those cells are not going to engraft. So what we needed to do is restrict that expression using the same system, but restrict the expression to the cells that are actually doing the job, which is the monocytes and macrophages. So very briefly, you may know Gaucher disease mostly as Gaucher disease type 1, a disease that manifests in kids with hepatosplenomegaly, failure to thrive, bone involvement. There are other forms. Gaucher is actually quite complicated. There are other forms that are known as the neuronopathic forms, which have neurological involvement, and there are sort of different degrees of how severely affected they are. Type 1 is usually thought as non-neuro involvement, unless you think of a 20-fold increase risk of getting Parkinson's disease involvement, which I do. So they have a really high risk for Parkinson's disease. So again, this enzyme is a different enzyme, it's glucocerebrosidase, but in the same sort of pattern. Without the enzyme, you accumulate substrate like glucosylceramide. And Gaucher, unlike MPS 1, the problem we think, at least in Gaucher disease type 1, is that you have these macrophages that get infiltrated with debris, and that is exactly what's causing all the symptoms. And they are signaling abnormally, there's weird inflammation, and the problem really is in the monocyte macrophage lineage. And these patients get enzyme replacement, and pretty cool, they also get a small molecule which reduces the substrate. Both of these work really well. And in the past, people used bone marrow transplantation, which was a one-time therapy for GD1 and is being used in neuronopathic forms when like ERT and SRT don't work. But again, given the risk of a bone marrow transplant, people said, these two work pretty well. We're never going to do this again. But now with the ability of actually engineering the patient's own cells and making sure that we can replace the defective lineage, which is the monocyte macrophages. We really have the opportunity to provide a safer one-time therapy for Gaucher. So this is where I was doing earlier. We are expressing enzymes everywhere. Here's what we're doing now. We use just a different promoter. So we use a promoter from a gene that is very specific to these cells. And so what happens is we don't get expression in the hematopoietic compartment, so we preserve engraftment. But so in the early cells, you don't see expression of the marker. But as the cells begin to differentiate in a macrophage, that's when you start to see expression. And so you can see that you can use this safe harbor system to actually play, not only just use it for enzymes, different mutations, but also using this ability to change promoters and to result in very specific patterns of expression. It actually can be quite customizable. And this is showing you that these cells can make healthy human macrophages, and we're trying to treat a human engraftable model of Gaucher disease. Now, I keep telling you that these transplants are awesome, but they really aren't always. And part of the reason is that they really don't treat cells, diseases that have rapid neurological decline very well. And that's because, you put a stem cell, it has to kind of engraft, and then it has to proliferate, it has to migrate. So really for transplant to be therapeutic in these diseases, it takes months. And that's part of the reason why people are so selective in choosing patients that are going to be transplanted. So to model diseases that have rapid decline, we're using Krabbe as a model, and the idea is you really don't have time for the cells to come from the bone marrow. You're going to have to deliver enzyme to the brain as quickly as possible. So this disease, again, is another lysosomal enzyme, galactocerebrosidase, and severely affected individuals, starts really early, and if you don't do anything, they rarely make it into their second year. And this is the enzyme that is needed for myelin turnover, both in the central and peripheral nervous system. So it's a bad neurological disease. So the idea here is to do the same, but then we have a couple other options. We can make monocytes and macrophages from this population that we already engineer and directly deliver it into the brain. And we also have other potential stem cells that can do this, and we're working with somebody else in engineering human neuronal stem cells, which in addition to making an enzyme, they can also have the ability to replace some of the cells that these patients are losing, which are the oligodendrocytes. And so we're trying this on our mouse model of Krabbe disease, which is known as it's fun to show you a video. So it's known as the Twitcher mouse, and I hope you can see why it's called the Twitcher mouse. So this is a summary of everything that I've told you about therapeutic genome editing and lysosomal storage diseases. So genome editing allows for precise genetic correction, as I mentioned before, but also the ability to synthesize new coding and regulatory sequences, and you can get very imaginative about how you can use this. The safe harbor approach that I've described is very flexible for the expression of, lysosomal enzymes, but you can, again, be more imaginative and think about all the therapeutic proteins that could be secreted from the bone marrow that would have therapy value or, other value, aging or whatever, aching joints or something like that, I don't know. So they're described in vivo and ex vivo genome editing approaches being developed for lysosomal storage diseases, and I think you guys are pretty up to date in what's happening in this field now. Hopefully you've learned that the hematopoietic system can be used to deliver proteins to other organs other than the bone marrow. This is a system that we have decades of experience using and modifying, so why not try to leverage everything that we know in order to treat these other diseases. These effector cells that we really care about are macrophages, and this, as I mentioned, is applicable not just to lysosomal enzymes, but other proteins that have therapeutic potential. And with that, I want to thank everyone. Harvey, my lab, which is the only gene repair shop in town, and all of the funding agencies, and Matt. Thank you.
Natalia will welcome some questions. Harvey.
It's the portrait mode. We talked about this yesterday.
That's fantastic work. Really honored to be associated in some way, because I remember taking nothing the potential of the future is great.
Right.
How would you think about protecting against that?
Yeah, so I mean, we kind of have to figure that out experimentally. When you use these, and what the promoter that we're using here is really just a short version of a endogenous promoter. So endogenous promoters are usually, tend to, I mean, unless you're using something like actin, they don't really express that much. For this version of this promoter, the cells only make twofold more than normal cells. So, I mean, the promoter itself will detect the amount, and experimentally you decide if it's, too much or too little. There's really no way to predict that. Okay, over here.
Does this sort of operation also offer the potential for creating super normal individuals, super athletes?
I think that's, if you use your imagination, you can come up with some things. I mean, can you think of a protein that you can have expressed in the blood that will, a lot of people are talking about circulating things in the blood that are, preventing aging, right? So people are getting transfusions from younger people. Is that something, a function that is encoded in a single protein that maybe you can put back in bone marrow? I mean, maybe. I'll have to think about something like that to get some money.
So do the ability for stem cells, and secondly, how much can actually be applied to the embryo?
Yeah. So a lot of the experiments that we're doing are derived from cord blood cells. I think that in the longer term, when these are going to be used in clinic, you probably need a lot more cells from the patient in order to get enough modified cells. So if you have adults, maybe you can mobilize them a few times. If you have babies, we've been talking about other ways to getting enough cells to do the modification. Because remember, right now it's not 100%. So we get enough for our experiments, but I think for the treatment, you're going to need more than what you get from cord blood. Maybe, unless we learn how to expand them, which people are working on this, and that would be great. If you can expand them, then that would be useful. In terms of, yes, you could, people are thinking about transplanting, well, generally using genome editing in general in utero. It's easier if you think about not transplanting cells, but just injecting the genome editing components, like the in vivo approach, and you just kind of hope they get to the right tissue and the right amount. But people are also thinking about doing hematopoietic stem cell transplantation in utero. It doesn't work great. Like the cells don't. You think that they would engraft, but they don't engraft fantastically well. So it needs some work, but it's definitely a real research idea that people are pursuing.
Do you have any evidence in your mouse models that age of the mice impact efficacy when you?
Absolutely. I mean, I haven't tried to do the correction on different, ages, but certainly the engraftment of the cells is better if they're younger. With MPS 1, for example, so I tell you that the mice have MPS 1, but mice don't care about hyaluronidase as much as we humans do. So the mice actually live a long time compared to a patient with severe disease. And so we actually kind of have to wait a long time for them to really manifest neurological problems. So all these things are time dependent. And yeah, I mean, technically the neurodegeneration is degeneration. So the idea is that the earlier you do it, the better they're going to do. Some of the things are reversible, like spleen and liver stuff, but bone and brain, you have a pretty tight window.
All right. Thank you very much.
Thank you.